Black is one of the most common coat colors in domestic cats. Walk into any shelter and black cats will be among the most numerous animals in the building. The color is so common in domestic populations that it barely registers as remarkable. In wild cats, the situation is almost the opposite. Among the 37 wild cat species that exist today, solid black coloration is a rarity. Most wild cats are spotted, striped, or tawny, with color patterns that serve as camouflage in their particular environment. When black does appear in a wild species, it stands out as unusual. Scientists looked at this discrepancy and asked a simple question: what is the genetic relationship between the black domestic cat and the black variants that appear in wild species? To answer it, they compared the domestic black cat's genetic mutation to the melanistic mutation in every wild cat species that carried one. Every hair on every mammal gets its color from the same basic process. Cells in the skin produce pigment and load it into growing hair follicles. Those cells have a default choice between two types of pigment: a dark pigment that produces black and brown tones, and a light pigment that produces orange, yellow, and red. Which pigment gets made depends on signals from other molecules in the cell. Two genes control most of that signaling in cats. One gene acts as an accelerator, pushing cells toward dark pigment production. The other acts as a brake, suppressing that signal and allowing light pigment to appear. Black coloration happens when the brake gene stops working, leaving the accelerator permanently active and the cell permanently producing dark pigment. The mutations that break the brake gene are what researchers compare when they map melanism across species. When researchers mapped melanism across all 37 wildcat species, a pattern emerged that changed the question from simple to complicated. Black coloration had evolved in the cat family not once or twice, but at least 13 separate times. 13 different mutations in different branches of the cat family tree, all producing black animals through related but distinct genetic changes. Some of the mutations were in the brake gene, some were in the accelerator gene, some were in regulatory regions that control how loudly either gene is expressed. The convergence was striking. The same visible outcome, black fur, kept appearing in different species through different genetic routes. The cat family seemed to keep rediscovering black coloration independently, which suggested that black carried some advantage worth finding repeatedly. 13 independent origins of the same visible trait is a striking number. It means the cat family has been solving the same problem over and over in different branches across millions of years of evolution. When a trait evolves that many times independently, it signals that the trait is solving a real problem, rather than appearing by random chance. Something about being black was useful enough that natural selection kept finding it in forest cats of Asia and Africa and the Americas, through whatever genetic route happened to be available in each particular species. The comparison of those routes, laid out across the cat family tree, showed a pattern in which the specific position of the mutation shifted from species to species, even as the outcome remained the same. The cat family kept arriving at black through different doors, which is why the domestic black cat's match with any wild species was never guaranteed to exist. The black leopard, called a black panther in popular usage, is one of the most recognized melanistic cats in the world. Its genome has been studied in detail, and the mutation responsible for its dark coloration sits in the brake gene. A specific sequence of changes in that gene disrupts the signal that normally holds dark pigment production in check. The mutation is recessive, meaning a leopard needs two copies, one from each parent, to be fully black. Leopards that carry one copy look spotted. Only those with two copies display the solid, dark coat. The mutation has been found in leopard populations across Asia and Africa, suggesting it spread through the population from a single origin point, and has persisted because black coloration offers a hunting advantage in dense, forested environments where spotted patterns provide less concealment than in open terrain. The black jaguar achieves its coloration through a mutation in a different gene entirely from the black leopard. Where the leopard's melanism involves the brake gene, the jaguar's involves the accelerator gene. A dominant mutation in that gene locks the accelerator in the on position, producing black coloration even in a jaguar that carries only one copy of the mutation. This means a single black jaguar parent is enough to produce black offspring, which is why black jaguars are more common in some populations than black leopards are in theirs. The genetic mechanism is different, the chromosomal location is different, the inheritance pattern is different, and yet the result looks the same to the eye. Two species, both called black panthers in casual usage, achieving the same appearance through entirely separate genetic paths. Among the smaller wildcat species, melanism appears in several lineages, and the genetic basis varies considerably from one to the next. The serval, a long-legged African species, produces occasional melanistic individuals, whose coloration traces to a mutation in the brake gene, but at a different position than the leopard's mutation. The oncilla, a small, spotted cat from South America, shows melanism through yet another variant, and researchers studying South American small cat genetics found evidence that the oncilla's melanistic mutation had spread into related species through interbreeding, carried from one species into another along the genetic borders where populations overlapped. The jaguarundi, an unusual Central and South American cat that naturally comes in dark, unspotted forms without needing a mutation, represents a case where the color pathway was redirected at an earlier point in the species' evolution, producing dark coloration as the default rather than as a variant. The variation in inheritance patterns across species adds another layer to the comparison. The leopard's melanistic mutation is recessive, requiring 2 copies to produce black coloration. The jaguar's is dominant, requiring only 1. Among the smaller cat species, both patterns appear in different lineages. A recessive mutation spreads slowly through a population because it can hide in carriers who show no visible sign of it, and only surfaces in animals that receive a copy from both parents. A dominant mutation spreads faster because it shows up in every animal that carries it. The domestic cat's melanistic mutation is recessive, which is why solid black domestic cats have 2 black parents, or parents that are carriers. Running the inheritance pattern comparison alongside the genetic sequence comparison gave researchers additional data for distinguishing which wildcat mutations were truly similar to the domestic cat mutation, and which only resembled it in the final visible outcome. When researchers plotted the geographic distribution of melanistic wild cats, a pattern emerged. Black variants appear most frequently in humid, densely forested environments. Melanistic leopards are more common in the rainforests of Malaysia than on the open savannas of Africa. Black jaguars concentrate in the dense interior of the Amazon basin rather than in drier forest margins. The serval's melanistic populations cluster in highland forest zones of East Africa. The pattern is consistent enough that researchers proposed a straightforward explanation. In dense forest, where dappled light and deep shadow replace the open visibility of savanna, solid dark coloration may outperform spotted patterns for an ambush predator. The same spotted coat that hides a leopard on a sunlit savanna makes it visible in a forest where a solid, dark animal would simply disappear into shadow. Black coloration got selected for in forested environments and eliminated in open ones, which is why it kept reappearing wherever the forest was. With a catalog of wildcat melanistic mutations assembled, the team turned to the domestic black cat. The domestic cat's black coloration also traces to the brake gene, the same gene responsible for melanism in leopards and several other species. The specific mutation in domestic black cats had been identified previously and was well characterized. The team placed it alongside the wildcat mutations and ran the comparison. Which wildcat's melanistic mutation was most similar to the domestic cats in terms of the specific position in the gene, the nature of the change, and the downstream effects on how the gene functions. Most of the wildcat mutations came back with clear differences. The Leopard's mutation sat in a different position. The Serval's variant differed in the way it disrupted gene function. Several of the South American small cat mutations clustered together but away from the domestic cat. Then the comparison reached the African golden cat. The African golden cat is not a widely known species. It lives in the rainforests of Central and West Africa, rarely photographed and poorly studied compared to larger or more charismatic cats. It's roughly twice the size of a domestic cat, with a coat that ranges from golden brown to reddish gray depending on the individual. Melanistic African golden cats are common, more common than melanistic variants in most other wildcat species, with some forest populations showing black individuals at rates above 50%. When the comparison reached the African golden cat's melanistic mutation and placed it alongside the domestic black cat's mutation, the genetic distance was noticeably smaller than for every other wildcat tested. The specific changes in the break gene in African golden cats and in domestic black cats sat at closely related positions, disrupted the gene's function through a similar mechanism, and produced proteins that functioned comparably. Of all the wild cats with black coloration, only the African golden cat matched. The similarity between the domestic black cat mutation and the African golden cat mutation raised an immediate question. What does that match actually mean? The African golden cat lives in the same general region of the world where domestic cats originated. The ancestral wildcat that gave rise to all domestic cats lived in the Middle East and North Africa, which puts the domestic cat's evolutionary origin geographically close to the African golden cat's range. Whether there was any ancient contact between the lineages that could explain the similar mutation is an open question. One possibility is that the similar mutations arose independently in the two species through convergent evolution, the same position in the same gene changing in the same way because that particular change is genetically the most accessible route to black coloration. Another possibility is that the mutation has a common origin much further back in the shared ancestral cat lineage, predating the split between the domestic cat's ancestors and the African golden cat's ancestors. The gene responsible for black coloration in cats does more than determine fur color. The break gene that produces melanism when mutated is part of a signaling pathway that operates in multiple tissues, including cells of the immune system. Researchers studying the relationship between coat color and disease resistance in mammals found that the same signaling pathway plays a role in how immune cells respond to certain pathogens. Melanistic animals in several species, including cats, show measurable differences in immune response compared to their non-melanistic counterparts. In domestic cats, black individuals have been found in some studies to show greater resistance to feline immunodeficiency virus, the cat equivalent of HIV. The resistance isn't absolute, and the evidence is not definitive enough to make a clinical claim. But the genetic mechanism connecting black coloration to immune function is real and stems from the shared pathway that the break gene operates in. The African golden cat shows a parallel pattern. In field surveys and captive populations, melanistic African golden cats appear to show lower rates of certain parasitic infections than golden-coated individuals from the same populations. The sample sizes are small and the data is preliminary, but the trend aligns with what the immune gene pathway comparison would predict. If both the domestic black cat and the melanistic African golden cat carry mutations in the same position of the same gene, and that gene influences immune function as well as pigmentation, then the immune effects of the mutations should be similar in both species. The comparison of black cat DNA to wild cats started as a question about color. The immune pathway connection turned it into a question about disease resistance as well. Black coloration in domestic cats carries a statistical footnote that connects to the shelter populations where black cats are common. Studies consistently find that black cats are adopted from shelters at lower rates than cats of other colors, a phenomenon sometimes called black cat bias. The reasons offered are various. Black cats are harder to photograph, they look less distinctive in small enclosures, and cultural associations with black cats vary across different communities. What the genome comparison adds to this observation is a possible biological context. If black domestic cats carry a version of the immune-related gene pathway that confers some resistance advantage, and if that same gene pathway has been independently favored in wild forest environments across multiple continents, then black coloration in cats is not simply an aesthetic variant. It's a color with a functional history, selected for in environments where the advantages it carried outweighed the costs of being visible. The shelter statistics sit in an interesting relationship with the genetic findings. If black cats carry an immune advantage through the gene pathway that the comparison identified, and if that advantage is heritable and real, then the low adoption rates for black cats represent something beyond an aesthetic preference. The most genetically resilient color variant in the domestic cat population is systematically the last to find homes. Researchers who study shelter population dynamics have noted that black cats also tend to have longer shelter stays, which exposes them to the stress-related health effects of confinement for longer periods. The irony is measurable. The color that may carry the strongest biological advantage against certain diseases is the color that spends the most time in the environment most likely to compromise the immune system through chronic stress. The domestic black cat's place in this picture is unusual. Domestic cats are not forest animals. They live in human homes, in urban environments, in agricultural settings, none of which are the dense, humid forests where black coloration has repeatedly been selected in wild cats. Yet black is common in domestic populations in a way it never was in wild ones. The explanation likely lies in what domestication removed: predation pressure. Wild black cats in open environments get selected against because they're visible to larger predators and less effective in open terrain camouflage. Domestic cats face no equivalent pressure. The mutation that would have been eliminated from an open country wild cat population could persist and spread in a domestic population where visibility to predators wasn't a survival factor. The color accumulated in domestic cats by default, not by selection, while in wild cats it remained confined to the environments where it actually helped. Comparing domestic black cat DNA to every wild cat produced a result that ran in two directions at once. The color comparison showed that black evolved 13 times across the cat family through distinct genetic routes, and that only one wild cat's melanistic mutation closely resembles the domestic cat's. The immune comparison showed that the gene pathway responsible for black coloration influences disease resistance in ways that are similar across the species that share the closest mutation variants. And the geographic and ecological comparison showed that black coloration follows a consistent logic in wild cats, appearing in forests and disappearing in open terrain, while accumulating in domestic cats through a completely different mechanism that has nothing to do with environment and everything to do with the removal of the selection pressures that would otherwise have kept it rare. The African golden cat lives in the forests of Central Africa, rarely seen and rarely studied, producing black offspring at rates that make it the most melanistic wild cat species by proportion. The domestic black cat lives in shelters and apartments across the world, the most common color variant in the global domestic population, adopted at lower rates than its litter mates. The two animals are not closely related. Their shared mutation appears to be an independent arrival at the same genetic solution, or a very ancient inheritance from a common ancestor that lived long before either species existed in its current form. Either way, the comparison found the connection. Out of 37 wild cat species and 13 separate evolutionary routes to black coloration, only one came back matching the domestic cat's version of the mutation. The rest were close enough to recognize as family and different enough to be unmistakably distinct.